excited today to have one of our own students speak to you about research that he did this summer in cooperation with the Untitled Institute. So if you're a student in biology, you can take courses there. And Nick was able to take courses there last year and then go back this year to do research. So he's going to be talking a little bit about some of his work with macroinvertebrates, which we'll explain to you, on the Borden River and the consequences of building dams and rivers. So without further ado, I'll just let Nick take it over.
Can everybody hear me? So as Professor Wickland mentioned, I'm Nick Vanderstelt from Spring Arbor University, and then I worked on this project in concert with my friend from college at Nazarene University, Derek Iwema, and he is in California right now, so he can't be here to present with me, but I did this research presentation and project through the All Institute, and it was through a internship that I was able to, and research position that I was able to complete with them this summer. So that was the 2018 River Dams Macroinvertebrate Study. So this is an ongoing study that was begun in 2008, and it was continued all the way to this past year and will be continued in the future as well. It started with 2 sites, and it's expanded all the way. This year we did 14 sites, which is quite a lot to handle, as you'll come to know later. The Borden River is just over 28 miles long and flows from Kalkaska County to Grand Traverse County and empties into the west arm of Grand Traverse Bay. This star right here is Cedar Bend Farms, so you guys are all familiar with that, and that's just kind of putting it in context for you. So you know that the water's really cold up there, and I also know that because I was in the water a lot. The watershed is 295 square miles, and all of that water is pulled into the Borden River and drains into the Grand Traverse Bay. These are 2 typical images of the Borden River. The one over here is after a dam has been removed. As you can see, there's a lack of trees in this section here, and that's because this whole section was underwater before, after and before the dam is removed, but now it's able to be restored. And this is a more typical image of what the Borden River would look like. As you can see, the banks are lined by mature trees. That's an area known as the riparian vegetation. And that's very typical of the Borden River. Most of it is surrounded by mature forest. There's very little agriculture up there, which provides a good buffer for the stream for pollutants, as well as it absorbs a lot of the rainfall, so you don't have a lot of fluctuation in the water levels. So this is another aerial view of the Borden River, and the red blocks represent the 4 dams that were placed on the river. The Brown Bridge Dam is the furthest upstream and was removed in 2012. The Boardman Dam was removed in 2017. Sabin Dam was actually just removed a couple weeks ago. And then the Union Street Dam is downtown Traverse City. If you've ever been downtown Traverse City, you've probably seen the Union Street Dam. It's a very popular area for people to kayak and just navigate throughout the Bay Area. And the Union Street Dam is actually scheduled to be renovated in 2020. It's not actually going to be removed because it acts as a lamprey trap and a barrier from the lamprey to enter from the Great Lakes into the Boardman River. And the Borden River is the reason why that's important is the Borden River is a cold water blue ribbon trout stream, which attracts anglers from all over the globe, myself included. I didn't have to go that far, which is nice. So this is actually a downstream view of the Sabin Dam. This is a picture that we took this summer. As you can see, there's quite a bit of current flowing through the spillway here, and then the gate here can be opened and closed. And all of these dams were put in place as they are useful to use for hydroelectricity. So the reason why they're put in place is while logging and industry was going on, they were put in place about 100 years ago in order to generate revenue, and they're kind of like a renewable resource or a green energy. However, as the structures degrade and time goes on, more and more money is put into upkeeping these dams as a structural integrity, withstanding thousands and thousands of gallons of water each second even pouring through, as the discharge is quite large, really wears on the dam structure. And there's several $1 million that are put in each year to upkeep these dams. And as of about 30 years ago, less and less hydroelectricity is generated because of sediments that impound on the backside of the dam as well. which is one of the drawbacks. So as the dam ages, the structural integrity is compromised, as well as sediment is impounded on just above the, this is called the impoundment, or it's a berm, is also known as a berm. And the more sediment that impounds here, the less flow you get and the less hydroelectricity is generated. So the past 20 years, there really hasn't been very much hydroelectricity generated, and there's been millions of dollars being poured into making sure that this dam doesn't break, and it's not producing any money. So that kind of leaves us with what do we do with the dams when they should be removed. More ecological impacts of dams that are negative is debris and nutrients as they're swept down from upstream to downstream. That's kind of like the lifeblood of the stream. Other than algae, that's the main source of nutrients and food, and it's kind of like the base of the food chain. So without the debris and nutrients, the downstream sections of the stream is really robbed by dams, so that is one impact. So the debris tend to jam up and impound and get caught in the sediments, and then the organisms below the dam don't see the benefit of the organic input. Additionally, all these sediments that are impounded below the dam can get washed over the dam and cover the river bottom on the downstream section. This is known as the cobble, and that's hard rocks that line the bottom of the substrate of the stream. And this is important because many species rely on this hard bottom to grow, such as aquatic plants and macroinvertebrates, which I'll talk about in a bit. But if the bottom of the stream is completely covered with sediment, no aquatic plants can grow and very few macroinvertebrates can survive. Therefore, no fish, and you have an ecosystem which is suffering. On the bottom side of the dam, you have increased flow as water comes over, and it tends to degrade the riverbed there as well. So once you want to remove a dam, how do you do it? The above stream section is known as the Relic Pond, above the impoundment here, which stops the water from going up. And as water sits in this relic pond, increased evaporation and temperature increase occurs. This is important because a cold water stream relies on cool water to have high dissolved oxygen, and with all that dissolved oxygen, fish species and macroinvertebrates wouldn't be able to survive, and then usually that would just change the ecosystem drastically. This is a picture of the Boardman Dam, which was removed in 2017. So the first order of business when you're removing a dam is to draw down the water from the relic pond down to the downstream section. And this is a siphoning drawdown technique where pumps initially pump water from the relic channel or the relic pond up into tubes, and then let gravity and the properties of water take over from there. So this is a way to control the amount of water from the upstream to the downstream section. And this is nice because it slows down the flow and really mitigates the environmental impacts. This was not done on the Boardman Dam or the Brown Ridge Dam in 2012, as the pumps failed and the berm broke, and that just caused a lot of environmental havoc, and there was a lot of sediment that washed down uncontrolled. But this siphon drawdown technique is what they actually used in the Zealand Dam removal a couple weeks ago, and it's just a very safe and controlled method of removing water from the top of the dam to the bottom of the dam safely. This is a picture of that same dam that we took this year. So this impoundment here is what is turned into this road here. And as you can see, the original riverbed. So the U.S. Army Corps of Engineers are the ones that got the bid to do this job, and they took soil cores. And as they core the soil, they're hitting sand, sand, sand. There's a lot of sand in northern Michigan, if you've ever been there. And then they finally hit some stumps, which is the original riverbed, and then gravel, and then stumps again, which that's how they figure out where the original riverbed is, and that's called the Relic Channel, and I'll refer to that a little bit more as we go up throughout the presentation. But the Relic Channel, which is the original stream bed before the dam was put in place, is reestablished, and then the banks have to be stabilized, as you can see in the next picture. This is the Brown Ridge Dam, which was removed in 2012, and as you can see, secession is taking place here, and growth is reestablishing, which helps stabilize the banks and ensure that erosion and sediments aren't transported downstream. So now that the dams are removed and the process of dam removal is occurring, we have to monitor this. So as you remember, the red blocks represent the 4 dams, and these green circles all represent the sampling sites that we took. So we sampled just below the dams, as well as just above the dams in the Relic Channel. So typically, so for example, in this one, these all three were underwater underneath the Relic Pond of the Boardman Dam, and that's true of all the ones that are just above each red block. This green star here is Grasshopper Upper, which is our reference site. Now this site is far enough upstream not to be affected by dams, even the Brown Bridge Dam, which was moved in 2012. So this site is completely unaffected by this dam, as the flow rates are the same here, and the temperatures of the water remain the same as they have been throughout the past few hundred years. So at each of these sites, we want to so we figure out the sites that we want to sample. We want to sample right around the dams, and we're looking for organisms to study. And macroinvertebrates are anything that you can see with your naked eye that don't have vertebrae, and they can actually be very, very small. So the smallest end is smaller than a millimeter, and they can be all the way up to a couple centimeters in length. The average size of the ones we were looking at were just a couple millimeters, so we had to use dissecting scope to see all of them. And then this is the macroinvertebrate Bible. Has all of the macroinvertebrates of North America, and this was my best friend. I was hoping my wading boots and my waders would be my best friend this summer, but my dissecting scope and macroinvertebrate book ended up taking 40 hours a week for 10 weeks. So macroinvertebrates are good bioindicators, and bioindicators are species that help you make an inference about what's going on in the environment around them. So bioindicator species are helpful because they give us clues as to what is going on. So macroinvertebrates are important because they have low mobility in the streams. They can't move upstream very fast because the current is very strong. They also have a predictable and known life cycle. So as they hatch or as they are present in the stream, it's easy to know and identify where they're going to be. So that helps on when to sample and where to sample. And these three here are known as the EPTs, Ephemeroptera, which are the mayflies. It's the E. P is Plecoptera and the stoneflies, and T is Trichoptera, which is the caddisflies. And the Plecoptera are kind of like the holy grail of the EPTs, and if you have stoneflies in your river system, then it is a very, very good river. Now we're going to talk about a not so nice macroinvertebrate. This is the New Zealand mud snail. It's a non-native invasive from, you guessed it, New Zealand. And as you can see, compared to this dime, they are very, very tiny. Even the juveniles look like small grains of sand, and to the naked eye and to somebody that doesn't know what a New Zealand mud snail looks like, it looks like another grain of sand. And that's why it's so easy to spread, because they can get stuck to the bottom of your wading boot. You can't really distinguish from what is a juvenile New Zealand mud snail and what is a grain of sand. Additionally, you can see here, this is an adult New Zealand mud snail, and their head can retract, and they have what is called an opercula. This allows them to resist drying out, so they can live outside of the water for about 72 hours. And then once they're re-submerged into a moist environment, then they're ready to reproduce, which they can do sexually and asexually, which is terrifying. Additionally, they can the same method that allows them not to dry out allows them to avoid measures that we would use to get rid of them, such as molluscicides, which are a chemical mix that kills most aquatic snails. However, these will just retract in their shells and they will be unaffected, as well as predators that try to eat them. If they end up being ingested by a fish, they'll curl up into their shells and pass right through the digestive system, and then get spit out on the other side, ready to keep going. So they're very, very tricky to get rid of. In Yellowstone National Parks, they've reached population densities up to 300,000 per square meter, which is essentially covering the entire substrate. They're not at those levels in the Borden River. They're about, we measured them this year at about 1,000 per square meter. So we're talking about 2 orders of magnitude of the difference between 1,000 and a couple 100,000. Anyway, so these are quite a nuisance, and they definitely affected our data. However, we're going to talk about a little bit how we collected these macroinvertebrates. So first, we used GPS coordinates for each of our 14 sites, which we have remained the same, at least for sites that we've continued to sample in. And once we got to the GPS coordinates, we'd stretch tape perpendicular to the stream, and we would begin sampling 1 meter from each bank with a Serger sampler, which is a box frame, which is a foot by a foot, or 30.5 centimeters. And then we used a 2 foot long or 60 centimeter 500 micron mesh net. And 500 microns was just allows very small particles of debris and sediments to flow through, but all of the detritus, which is the leaves and the broken down sticks and the macroinvertebrates, to remain in the net. So we would put the surface sampler perpendicular to the flow, and the stream would flow and move all of the disturbed macroinvertebrates into the net. So we'd use this single prong agitator. Essentially, it's a gardening tool welded to a metal pole, and we'd use that to disturb the substrate. We'd move that in a grid motion for 45 seconds, and then we'd take our hand and use a single hand to brush off the larger rocks in order to ensure that all the macroinvertebrates in that square foot area were being transported from the riverbed to our net. And then we would take them and transfer them into a bucket and then to a sieve, and we would use 90% ethanol with rose bangle, which is a stain that stains living tissues pink, which helps the researcher distinguish a brown macroinvertebrate from a brown piece of leaf or stick. It helps us see it better under the microscope, which turned out to be very helpful. Once we had them in mason jars, which is what we would collect them in, we would then take them back to the lab and transfer them into labeled Nalgene bottles, and then we would sort them, which was definitely the majority of the time. So we got all of our sampling done within the first week and a half, and then after that, it was all lab work. So if you want to go into the biology field and research, know that you'll have fun in the field, but then you have to get the work done in the lab. We would also take physical and chemical parameters at each site. I'm holding a flow meter here. We would take flow, discharge, average depth, pH, dissolved oxygen, conductivity, as well as estimate the substrate, so the cobble, the small rocks and pebbles on the bottom of the stream, as well as if there was wood or sand or periphyton, which is aquatic algae present. And this is just to help establish or maybe draw correlations between macroinvertebrates and their preferred habitat, in case that comes up in future studies. Once we got to the lab, we would sort each individual sample with a dissecting scope, right here with some gooseneck lamps and a grid petri dish. And once we got each of them down to, we sorted them to 14 families. Some of them were unknown, and then we moved them down to genus. Here, so here's a stonefly, but most of them are very small. I mean, these vials are only about that big. So some of the smallest New Zealand mud snails are, like I said, about a grain of sand, about the size of a grain of sand. And we were really, really excited when we got to find a macroinvertebrate that was this big. Just kind of helped us enjoy the experience in something different other than New Zealand mud snails. So we identified 6,880 macroinvertebrates down to genus, and that was the bulk of our research as we underwent sampling on 14 sites. So we identified just under 7,000 macroinvertebrates, and we still have to identify the mites and some of the aquatic worms, as they are a trick to identify. But out of these just under 7,000 macroinvertebrates, we had 28 families and 2 subfamilies. The 2 subfamilies were Chironomidae, which are the midges, and those are, you need an expert to identify them down even to family. So we struggled with that, but we were able to get it. So the relative abundance of all the macroinvertebrates that we collected, as you can see here, the New Zealand mud snails accounted for 42% of the population. And as a non-native invasive, that's worrisome, as they can compete with the EPT and the other organisms as they feed on algae, which is the base of the food chain there. And the other organisms represented 29%, and then the EPT, the very sensitive, good quality organisms, represented 29%. In a healthy stream, what you want is 29%, you want 50% EPT and 50% other or more EPT. However, this was offset by the New Zealand mud snails, but if you ignore the 42% New Zealand mud snails, you see that the split is perfect, 50-50 for EPT and other organisms, which it shows that, first of all, that there's a lot of New Zealand mud snails, but other than that, the populations are doing good, and the levels of EPT to other are still healthy. Any ratio above 50% is excellent for EPT to others. So, but this is just to show that the New Zealand mud snails are very, very prevalent, and how they also affected a lot of the data that we took. So based on all of the macroinvertebrates we identified down to genus, we were able to calculate quite a few indices. So I'll explain the graphs quickly. Each graph has the sites from upstream to downstream on the x-axis, GHU is our reference site, and Save Miffle was the last site we sampled on the downstream side. And then the y-axis will differ based on the diversity that we calculated. So the Shannon index of relative diversity is a nonlinear comparison, so you can't use it, you can't directly compare them. However, it's just kind of a baseline, and it's a very popular indice that's used in ecology just to compare different groups and see what the relative diversity is. As you can see in the Relic Channels again are the areas that have been underwater for 100 years before the dams were removed. And then the arrows each represent dams that removed. So this is the Brown Bridge Dam, which was removed in 2012, the Boardman Dam, which was removed in 2017, and then the Saving Dam, which was removed in just this couple weeks ago in 2018. So as you can see, the Relic Channels, which are the areas of concern, and just below the dams are hosting numbers that are kind of through the run of what we'd expect to see. They're not outliers in the data, which is exciting. You can look at another one, the effective number of species. This is more of a weighted average, and it is linear, so you can't directly compare them. So it's an effective number of species. So this is saying how many species are there approximately at each site. So if you look at GHL, it only has an effective number of species of 2, but at that site there were 80% New Zealand mud snails, so that's going to drive that ENS down quite a bit. And the New Zealand mud snails range to taking up to 80%, down to around 14 or 15% of the populations at each site, but that varied. But as I mentioned earlier, they represented 42% of the total population. As you can see here, the Relic Channels and just below the dam are performing about as well as they are in the rest of the run of the river, as you can see here. But the New Zealand mud snails affected the ENS acutely, as the high number of New Zealand mud snails would drive down the ENS significantly. The Sorensen similarity coefficient is comparing each downstream site of the reference site to the grasshopper upper reference site. So you'll notice there are only 13 bars on this graph because we're comparing them to the reference site. So the reference site would be 100% on this on the y-axis here. As you can see, the Relic Channels are in line with the rest of the stream, and just below and above the dams, the similarity is similar to the reference site, which is exciting because that means that the macroinvertebrate communities are similar to the site unaffected by dam removal. You'll also notice that there seems to be a bit of a negative trend as you move from upstream to downstream, and this can be explained by the river continuum concept, which states that as a river goes from upstream to downstream, more water is present in the system, which allows for different habitats and different species. So that can explain that bit of a negative correlation there. Lastly, let's talk about the Hovenhoff biotic index. The previous have been quantitative indices, and this is more of a qualitative index. So this is based on tolerance values from 0. It's a weighted tolerance value, so it's the number of organisms and multiplied by their tolerance value. So a tolerance value of 0 is very, very sensitive, which is what you want. You want macroinvertebrates that are very, very sensitive, and then the 10 is the most tolerant value. And as you can see here, GHL, which is the most affected by the New Zealand mud snails, have the highest HBI. But as you can see, the Relic Channels are showing numbers that are about even with the rest of the sites. Even it's nice to see that even the Relic Channel, although the HBI is quite high, that dam was just removed last year, and that it's still being populated by macroinvertebrates that are sensitive. And as I mentioned earlier, the New Zealand mud snails are the highest in BDU and in GHL, and the New Zealand mud snails really affected a lot of our data, especially because the tolerance value of those are so high and they were so prevalent in the data. But the HBI was a good index to see the relative health of the stream, and the amount of macroinvertebrates that were present were very encouraging. So again, the relative abundance of the New Zealand mud snail is concerning. However, they're not quite as high as they are in Yellowstone, as they're actually several hundred thousand less than they are in Yellowstone, and they're not too much of a concern now. However, if they continue to grow exponentially, that could definitely be a problem for the Borden River. So then we're looking to answer the question, how is macroinvertebrate community responded to dam removal. Our findings show that the macroinvertebrates are quickly reestablishing themselves at sites even when they have been underwater for 100 years or the dam has just been removed. It'll be interesting to see how they responded to the Sabin Dam removal next year, but that's for next year's team to figure out. And macroinvertebrates communities are also similar across all the sites sampled, which is exciting as well because we identified, or we can say that they are reestablishing, but then they're also reestablishing in the populations that matter. So then the EPTs are able to reestablish even in a short time, which means that the environmental impacts from the dam removal is small enough where they're able to adjust to that stress of sediment and habitat change. Recommendation for future studies would be to have additional sampling sites below the Sabin Dam and the Boardman Dam, as we only had one site below the Sabin Dam and one site below the Boardman Dam. So additional sample sites would help us be more confident in the data that we're finding, in addition just to give us a better picture of the river overall. Additionally, to continue to standardize methods in 2017 and 16 and 17, the Great Lakes Conservancy actually got the bid for this job, so there were 2 years where the El Salvo Institute didn't do it, and they changed their methods a bit, which is tough to compare data from previous years to future years. So we would just stress that even if El Salvo does not get the grant to continue this research, that the methods are standardized in order to compare data, and also just to continue on to have data to reference for the New Zealand mud snails. Additionally, considering to sample in other seasons such as the winter would just give us a more holistic view of how the river is doing throughout the year. I'd like to thank Spring Arbor University, first of all, for helping me get this position. I wouldn't have been able to do it without all the help from the faculty and the staff here. Also for the Conservation Resource Alliance are the ones who provided the grant for the El Salvo Institute to do the study, and they also laid out all of the methods and the indices that we should calculate, as well as all of the other nitty-gritty details that goes into grant writing. The El Salvo Institute of Environmental Studies for providing food and lodging and a place to and workspace. I'd like to thank Dr. Brian Keys, who is my boss and is the director of environmental education there at El Salvo. And he was very, very helpful in educating me on macroinvertebrates. Dr. David Mahan, he used to be the head of this study from about 2008 to 2012, so he was very helpful in giving us tips and tricks to make sorting go faster and how to sample properly, as well as Dr. Fred Van Dyke, who is the head of at El Salvo, for continued encouragement and help in paper writing, grant writing, and also making presentation. And also Mr. John Pearl, who is the food staff at El Salvo, and he makes excellent double chocolate cookies, which we're enjoying here on our break after sampling. So I'd like to open it up for any questions, and thank you guys so much for listening.
I think you've done a great job on this whole damn project. If the river hadn't been so healthy, what would you do to fix it?
As far as the dams?
No, I mean, if you found, let's say after removing the dam, you found
Okay.
the negative impact.
Yeah, so
what would you recommend to fix it?
I would recommend the drawdown technique that was used for the Brown Bridge Dam in 2012. It wasn't done by the U.S. Army Corps of Engineers. It was done by another organization that bid lower than the U.S. Army Corps of Engineers. And after it went wrong, they kind of figured out that they should give it to the people that knew that what they were doing. Whether it was an accident, they didn't mean to have the dam break. But I would say using the siphoning drawdown to remove the water from above the dam to below the dam is the most controlled method and isn't determinant on pumps working or not working, as well as making sure that sediment traps are established below the dam in order to, because there's going to be sediment that's impounded above the dam and that's got to get trapped somewhere. So dredging both beneath the dam, making pits that the sediment can flow into and then be removed. That's another way that can be helped, as well as stabilizing the banks of the river after dam removal in order to prevent sediment from blanketing the substrate. That's what I would say. Yeah. Right.
you measure pH
Yep.
up and down. It seems to me with all that sediment, a lot of that's going to be leaf litter. It's going to have a lot of tannic acid in it.
Yeah.
You release all that acid, that's going to play havoc with pH.
Yeah.
Did you see pH differences between those dams that have been removed recently and those that haven't?
We didn't see a huge jump in pH. We measured pH on both, on 2 different probes, just to ensure that pH was accurate. And I have all the data in the spreadsheet. We can take a look at it, but there wasn't a significant difference in pH, like just directly below even Saban Dam, which actually when we were sampling, they released a bunch of sediment as we were sampling, and the pH didn't change much even from releasing that. And I'm guessing the tannic acid would also be leached out of, even though the dam is present. I'm guessing like the leaching process would continue even though the sediment is impounded there. Dr. K.
Any chance you guys found a new species?
I doubt it.
7,000 and no new species.
No.
That's impressive.
Yeah, I would agree. Yeah, this macroinvertebrate key has heaps of macroinvertebrates that we didn't find, and this is just one of the keys that we used, so there's more like it. Richard.
Do you think the species would be different if you sampled in different seasons?
So certain species would be absent. So when they hatch out, so most species are going to hatch either in the spring or in the fall, but they're all going to be present before, just after winter ends and spring begins. So the first hatch, the mayflies, the caddisflies will hatch out, and the stoneflies as well, but then there'll also be an additional species, separate genus that have hatched in the fall. But all of them will be present in the winter, as all of their life stages overlap, whereas they'll all be in their larval stages during the winter. Richard?
You mentioned a number of techniques that didn't work very effectively for dealing with the New Zealand mud snail. Do you
Yep.
know of any efforts that are showing progress?
Native parasites in New Zealand, but so they rely on a lot of hookworms and roundworms, which are parasitic to New Zealand mud snail, and those are effective in New Zealand, but it's kind of hard to introduce parasites that small into a river system successfully. There's a big debate on whether or not the New Zealand mud snails in the Borden River are the lake strain or the river strain, and that may have an impact on how fast their populations are reproducing. So there are New Zealand mud snails present also in Lake Michigan, but they're very small and they take a backseat to the zebra mussels and quagga mussels. And the gobies. A lot of invasive species and creatures. Any more questions?
You looked at a lot of other parameters as well in your studies. Is there anything you can seeing if those parameters are influencing the communities and the diversity, or are you just looking at dams?
Yeah, so our grant was basically just to study the dam removal, and we do take stock. They haven't always taken the physical parameters at each site, and so there have been some years where they have and some years that we haven't, and Dr. Brian Keyes is really working on making sure that that's done every year, so that because we're out there already collecting data, so we might as well take more physical parameters in order to see. That's part of the reason why we also took account of the substrate. So we were trying to see if the New Zealand mud snails preferred certain, maybe if they preferred certain temperatures, certain pHs, certain substrate type. Like, do they like rocks? Do they like sand? What type of, or what size of, or what class cobble do they like, etc. So even though that's not specifically what our grant was written for, we were trying to collect data on that as well.
So is there any possible future research looking at algae communities or the vertebrate communities?
Yeah, I think that would definitely be something that we could write a grant for. I think the reason why this got funding is it was started by Trout Unlimited, was the ones who originally funded it. Go figure. And that was also continued by the Great Lakes Environmental Conservancy. So I think that if we can get people involved and interested, and to research more about the New Zealand mud snail as an invasive species, I think that's probably the best bet for doing and for future research on the Borden River. All right. Thank you guys so much for listening.